access it later. Exactly. We have the pleasure of having today with us Yaquan Chang. Yaquan is a postdoc fellow at the ETH Switzerland, where she studies macroecology, functional ecology, and plant biogeography. And today she will present her results on tectono-geomorphic controls on plant biodiversity in the Hengduan Mountains. Whenever you want, Yaquan.
Okay, thank you very much to organizers for inviting me. It's a great pleasure to be here. So as introduced, oh, sorry, can I switch? Oh, yeah. So as I've been introduced, I'm a biologist in the training, but in the past few years, I'm kind of hanging between these disciplines and think all the time that do plants care about geology? Well, it's such a big question, and I still don't have a very clear answer. But I'm pretty being convinced that geology really matters. And also, I'm not alone in this journey. Now, more and more people from the ecology and the geology side trying to bridge in these disciplines and to look at tectonic and geomorphic processes to understand our current biodiversity patterns. And so far, a lot of organisms has been used in this type of studies, include plants, birds, mammals, amphibians, and fish. Well, it's a very exciting new emerging field, but actually this field exists even before these disciplines breaks around 200 years ago. So here, I would like to introduce one naturalist called Alexander von Humboldt. So he is a German naturalist, and he started his expeditions, traveled to the Americas to look at all these species living there. But unlike other naturalists in that time, he was not a big fan of collecting identified species and collect the specimens. Instead, he just travel the place and look at the plants and think, why you are living here? So in 1802, he climbed a big mountain called Chimborazo in Ecuador. And in this expedition, he recorded very detailed plant information when climbing the mountains. And also he recorded the surrounding environmental conditions, like the elevation, temperature, air pressure, and etc. And after assembling these informations, he realized these organisms are not deadly aggregated. Actually, everything is connected. And he asked this fundamental question in biogeography is what grows where and why? So after centuries of data collection and technology development, nowadays we are be able to make a very high resolution biodiversity map. And in this map, and this is specifically for the plant species. In this map, we could see that the biodiversity pattern is not evenly distributed. Some places has extremely diverse plant species, while some places are is species poor. And when we overlay with the mountain regions, we could find the most biodiversity hotspots are for in these mountain ranges. And why is that? So after going into more detailed mechanisms, I would like to start clarify some biodiversity concept, because I realize that sometimes we use it in a kind of loose way and maybe confusing people from other disciplines. So in general, biodiversity is the number of species occurring in certain place, and we call it as the richness. And the biodiversity also has different components. For instance, it could be split into alpha diversity, which means the species richness at plot level. Imagine you just go to the field, go to the mountains, and to make a plot, maybe 20 by 20 meter, and identify all the species you could find. then the number of species you found in that plot is the alpha diversity. And the beta diversity is a change of species between plots. So it is a pairwise dissimilarity matrix to say how similar your species are when crossing time, crossing scale, crossing spatial scale, and it could be also crossing time. And also the gamma diversity that represent a regional level richness. Imagine you sample the many plants in these mountains, you could get how many species you could find in these mountains. And that is actually the gamma diversities. And using these metrics, we could understand some general process in driving the biodiversity patterns. So one relationship is the species area relationship, which is very intuitive. If we sample larger places, we could have higher probability to find more species. And also with the species productivity relationship. So productivity here means resources, such as food for animals. So if a place with higher productivity, that means it can has potential to host larger populations, and then it will end up in higher diversity due to the higher mutation rate and speciation rate. And the last one is a richness heterogeneity relationship. So if a place with very different environment to coexist, it can allow more different species to coexist, and that will drive high beta diversity, and then finally ends up in high gamma diversity. However, when we use these mechanisms to build a model, we could find large residues left in the world. And that means there are a lot of variations still cannot be explained by these mechanisms I mentioned above. And the positive residues are actually mainly concentrated in the mountain region. Therefore, we may need to look at the geological mechanisms. So here, one mechanism I'm going to mention here, we call it as a habitat creation. It means the tectonic uplift and the erosion process may create novel habitats for species to colonize and diversify. One example here is in the Tibet-Himalaya Hengduan region. So due to the collision of Eurasia and the Indian plate, this region experienced intensive uplift since 40 million years ago. And the biologists can reconstruct the evolutionary history of alpine clays in this region and found the high speciation rate period is associated with this intensive uplift rate. So we could conclude that the uplift is one of the main drivers for the Tibet Himalaya Hengduan regions flora diversity. And another mechanism I want to mention here is called habitat disruption. So it actually means the formation of the geographic barriers may impede the species dispersal, and therefore, due to this geographic isolation, the allopatric speciation will happen eventually. So, for instance, maybe some faulting processes can create such geographic barriers. And also maybe the river capture processes maybe split the one large population into several segments and result in high speciation. And here, one case study is in the Appalachian Mountains. Here, they found the formation of barriers through the lithology differences can create the allopatric divergence. They use the landscape evolution model to look at so they first their hypothesis is the sedimentary rock could act as dispersal barriers for these fish to migrate. And then they use the landscape evolution model to map to evolution history of these tributaries to see when the order of their geographic oscillations. And finally, they use some genetic information to build up the evolution history of this species and find the typology of the genetic information is very much consistent with the landscape evolutions. And another study I want to mention here is in the east coast of Madagascar. Here, due to the escarpment processes, the interfluvial ridges developed within each elevation band, and these interfluvial ridges can act as dispersal barriers for plants and create a high turnover within each elevation band. So if we want to work on this type of study, we first need to have very high resolution biodiversity data. So one way to collect the biodiversity data is from a community survey, like Humboldt did 200 years ago. So we just go to the forest and then we make a plot and to identify all species within this plot. It's a very precise and precious data, but as here you can see in this global map, it's very much unevenly distributed. And the most data are collected in the Europe, US, and Australia. Therefore, other ways of data collection are also being developed. One way is from the citizen science. So there are many apps. You can just download the app and take a picture of organisms you want to know what they are, and then you can upload your observations as well as geographic locations of these organisms. And finally, people can assemble all these data and to make this biodiversity map. This is an example of all the species observed by citizen science in the US. And another way to make a high resolution biodiversity data is from some statistical model. Here is a model called the species distribution model. It actually just uses species occurrence information as well as their surrounding environment to build some response curve to show how the occurrence probability varies along certain environmental gradient. And then you can reproject to the geographic space to see the potential distribution of certain species. And the last one I want to mention here is called the environmental DNA. It means you just get some samples in the environment, such as water, soil, or even air. Then you can extract the DNA from this sample and analyze it to get what kind of species you could find. I found this approach may be very interesting to geologists because if you sample the river eDNA, you will work on get the species composition at the catchment level. And I know many geological data also collected at the catchment level, such as cosmogenic nuclides. And apart from this biodiversity data, another data we need is called phylogeny. It's a time machine to help us travel to the past. And the phylogeny can have many different levels. For instance, for one species, we can get some intra-specific variation of this species to see how the population developed within this species. And we can also get a clade level phylogeny, for instance, one genus or one family, and to have their evolutionary history of these genus or family. Here I use an example of the rhododendron, and each tip here means a species. And of course, we can use all species in the world and build a mega tree. And this is an example from the angiosperm, and each tip here means a genus. So we can know the full evolutionary history of the angiosperm in the world. And by using this distribution and phylogeny, we can make some case studies And here, what I did is I use the Hengduan Mountain as a case study because it is a biodiversity hotspot outside of the tropics and with very complex geological history due to the Indian-Eurasia collision. And when we zoom into this region, we could find this region has very high elevation changes, and in the north it is has very high elevation with several mountains even over 6,000 meters, but in the south it has low elevation. And this region also has these very unique drainage patterns where three big rivers run in parallel with each other. And when we look at the tectonic faults of this region is actually dominated by the left lateral strike slip fault that indicate the whole region is experiencing the clockwise rotation. And that means the whole landscape has a lot of disrupting in the past and now. And from the lithology map, this region has very diverse lithologies and include a lot of bedrock types. And in the north, it's mainly dominated by the sedimentary rocks. And this region also has very unique climate information. So here I make a map for the growing degree. It means a measure of heat accumulation over a specific period. So it's a biological meaningful temperature proxy. And here we can see in the south, it has very high growing degrees, but in the north, it has very low growing degrees. And we can also make some proxy for the precipitation. And here I use the set water balance. So it is an estimate of water availability for species to grow. And here in both in the west and the east Hengduan region, we can see they have very high set water balance, and that's an indicator of the monsoons. And in this region, we could also use species distribution models to make a very high resolution biodiversity map to understand the specific mechanism in driving this pattern. Therefore, by using this information, we can ask how the tectonic geomorphic processes could impact the plant diversity in the Hengduan Mountains. First, we ask how does the habitat heterogeneity determine richness patterns in Eurasia and Hengduan scales? Why we start to understand the pattern at the Eurasia scale? Because like we know Hengduan region is a biodiversity hotspot because compared to other regions, other adjacent regions, it has much higher diversity. So we would like to know which family actually contribute to this biodiversity pattern. And here we consider the habitat heterogeneity is a first order process. And to quantify the habitat heterogeneity, we are using the growing degrees, the set water balance, and the bedrock types as different aspect of the habitat heterogeneity. And then we can reclassify them and make some unique combinations of these parameters. And each unique combination means a habitat type. Then we can calculate the heterogeneity matrix to say how the heterogeneity level looks like at the Eurasia scale. And therefore, we are be able to make these richness heterogeneity relationships. And we can do this family by families. And we know that they have a general trend with increasing of the habitat heterogeneity, the richness should increase. But if we make some quantile regressions, we can say like actually each dot here represent a region in Eurasia. So if we make the quantile regression, we can say not all regions follow this trend. Some regions can be identified as outlier region. And then we are just looking at which families in Hengduan Mountains could be identified as an outlier because we consider as the habitat heterogeneity is a first order process. If they beyond this expectation, that means tectonic geomorphic disruption may play a role in driving the biodiversity patterns. And then we could identify these outlier families at the phylogeny, and then here are two examples of the outlier and the non-outlier families. So the outlier families is Iraceae. So the rhododendron is in this family, and we can see it's highly concentrated in the Hengduan Mountains. And one non-outlier family here is Apiaceae. We call it as an umbrella family. Although in the Hengduan Mountains, it has a pretty high diversity, but the hotspot of this diversity is mainly concentrated in this region close to Iran. And after identifying these outlier families, we can accumulate these outlier families and make a even more detailed biodiversity map within the Hengduan Mountains. And we could identify the biodiversity hotspot with the contribution of these outlier families. And then we could to look at to what extent the habitat heterogeneity can explain these biodiversity patterns within the Hengduan Mountains. Here we found our heterogeneity matrix can explain half of the richness in the Hengduan Mountains. However, large residues still remain, and they have a very spatial clustering patterns, which may indicate some tectono-geomorphic disruption processes. Therefore, we could conclude the habitat heterogeneity explains the plant richness pattern, but the residues may indicate other complementary mechanisms. And secondly, I would like to zoom into the specific region to ask if mountain range can act as dispersal barrier for plant and fungal communities. Here we zoom into this the West Hengduan region, and this region has these three rivers run in parallel with each other. So our hypothesis is this big drainage divide between rivers can act as the dispersal barriers and to resulting in high richness in this region. Therefore, we did environmental DNA field work in this region. We select 30 drainage basins and then to sample the river eDNA from the outlet of each drainage basin and to analyze what kind of plant and fungal communities we could find in each drainage basins. And for all the genus we identified from environmental DNA, we can get their elevational ranges in this genus, and then we can make a histogram of in which elevation band they most likely occur, and then we compare with the elevation for the whole drainage basins, which shows the Gary density plot here. And when comparing the plant preferred elevation band as well as the basin elevation densities, we can see a consistent shift towards lower elevation. That means in this river eDNA approach, even though we sampled the outlet of the drainage basin, we are most likely to collect these mid to low elevation species. We are not be able to capture the full species in the whole drainage basins. So this, if we want to get to know all species within each drainage basins, we may be need to sample more tributaries within this drainage basin. And to have all these mid to low elevations, we could look at their beta diversities. So here I make a PCOA plot, and each if they have similar color in these drainage basins, that means they have more similar species. So here we can see a general trend between different river valleys. That means maybe the divide could act as a dispersal barrier. We further look at how the beta diversity distribute when crossing different rivers. And the blue line here means the elevations, and the pink bar means the beta diversity. Here we could more or less say, like, when crossing the drainage divide, it tends to have a higher beta diversity. When crossing river valleys, it may have lower beta diversity, but it's not always true. And another thing we need to think about is different river valleys maybe have different climate, and these climate variations can also filter different species in different river valleys. Therefore, we need to exclude the climate effect. So how could we do that? So here I would like to introduce the community assembly rule in ecology. So imagine we have a species pool. So all these species could live in certain place. However, due to the environmental variation in this place, and they may filter out some species. And for instance, maybe one river valley is very humid, it will filter out these dry tolerant species. And after that, due to some dispersal barriers, more species are not be able to get into the species. And finally, in the reality, what we observed is the community we observed is actually after these processes. And if we apply it in the mountain region, we could find maybe in the two communities in both sides of the mountain, they are pretty different. But on one side, they have a very high humidity, and another side they are very dry. And how can we know these community differences due to climate or the dispersal barrier? If we could manipulate the climate, then we could solve the problem. If we can manipulate the climate to let both sides of the drainage divide has very high humidity, then we could compare the community directly. If two communities are become similar after controlling the climate, that means this drainage divide is an ineffective dispersal barrier. And but if these communities crossing drainage divide are still different, even after controlling climate effect, that means the drainage divide play a role in impeding species dispersal. And how can we do that? We cannot manipulate nature, but we could do that in the statistical models. So here we could select communities within a river valley and to build a beta diversity purely due to climate difference. And then we can predict, using this model to predict the community's cross drainage divide to say how the beta diversity should look like if just climate play a role. And after that, we could use this predicted beta diversity to compare with the observed beta diversity. If the observed beta diversity is significantly higher than the predicted beta diversity, that means drainage divide is an effective barrier. And then we could say, we could make such conclusion. And if the observed beta diversity is fall in the predicted beta diversity, that means the drainage divide is an ineffective barrier. And when we apply this approach in the Hengduan region, here we could say, although we can say some species differentiation between different river valleys. After controlling climate, only Salween to Mekong Valley is an effective dispersal barrier. So these SM valleys. So that means for the plant community, the Salween-Mekong divide is a real dispersal barrier. Well, for the fungi communities, they show no differences between the observed and the predicted beta diversity. That means for the fungi community, none of the drainage divide acting as dispersal barrier. Therefore, we could conclude mountain ridge can act as dispersal barrier, but it is taxa dependent. After controlling climate effect, only plant communities shows exhibit high beta diversity across these mountain ridges. And the third, we asked how do tectonic and climatic factors influence the plant diversification histories? So there are many ways to quantify the diversification history. And here I would like to introduce a proxy called biogeographic realms. So it's a division of the landscape based on the species distribution information as well as the evolution information from phylogenies. Therefore, we can split the whole landscape into several subregions, and each region has more or less similar evolution history. And to understand what could drive these division patterns, we could use some climatic and geographic predictors. So one way to do that is we can randomly select points in adjacent realms and then to look at their climate difference as well as geographic isolation. And here is our result. So using this species distribution and phylogeny information, we are be able to classify the whole regions into 8 realms. And when we're using climatic and geographic isolation to explain this division pattern, we found for this north-south division, climatic variables plays a major role. Well, for the east-west division, the geographic isolation plays a major role. And as we know, in each realm, they have similar evolution history. We would also like to know if the tectonic history can also drive such pattern. So one way to do that is we could get some information from low temperature thermochronometry. So if the landscape is in the steady state, the erosion is always balanced with the tectonic forcing. But when the erosion changes in different time steps, that means the landscape regime changes. And that may mean either the landscape creating new habitats or disrupting existing habitats. And another way to look at it is the spatial variation of the erosion pattern, because these variations may create lateral movement of the drainage divide and therefore to disrupt existing habitats. And this is the exhumation rate result from my colleagues, and he built up this exhumation rate history from 30 MA until now, and to see how the erosion rate changes through time. And from the biology side, I could calculate the species age information. That means when the species speciate in the past. And here we could see in the east part, they have much more older species. And we can just make a very simple correlation between this age pattern as well as erosion complexities. So we could find the regions with longer deformation history, longer and complex deformation history will have more old species. Therefore, we could conclude climate and geographic isolation could jointly influence the division and the tectonic history, and the tectonic history is associated with the realm histories. And through these case studies I mentioned above, we might be able to summarize them to make a general approach to say how could we partition the biodiversity between environmental factors and the tectonic geomorphic disruptions. So first, we need to know what kind of process we are going to look at and construct the biodiversity patterns. For instance, if you want to mainly focus on the speciation processes, maybe you need to look at the richness pattern, or maybe or more precisely actually the endemism patterns. And then we could link this pattern with current or paleoclimate effect, because we know climate plays a very important role. And step 3, we need to find a way to account for these climatic effects. And finally, we can make a residual map to see what has been left after controlling the climate and trying to interpret the residual patterns with some tectono-geomorphic processes. For instance, here I use the catchment restricted relief as an example, but I know it's still a grand challenge and we still have a long way to go. Therefore, so to do this type of study, we first need to identify the pattern, account for the climate impacts, and finally explore the geomorphic impacts. So in conclusion, my work mainly emphasized the habitat heterogeneity is the first order process in driving species richness patterns, and the physical location of mountain ranges can act as dispersal barrier, but this barrier is taxa dependent and not all ranges are dispersal barriers. And also the climate and geographic isolation jointly influence the division, and the tectonic history is associated with the realm histories. So to better understand the geological drivers of biodiversity patterns, we first need to identify the pattern, account for the climate impacts, and then explore the geomorphic impacts. And finally, I would like to thank all the groups that have been involved in the past few years, and especially my supervisor, Loic Plessier, Sean Willett, and Niklaus Zimmermann for their support, and also many collaborators in China, in Switzerland, and also in the US. And of course, I also want to thank my cat. So his name is Huagang Yan, which means granite. And he really gives me a lot of motivation to learn more geology. Okay, that's it. Thank you so much. And I'm willing to take a question. Thanks.
Thank you very much, Yaquan. It's so super interesting. I'm sure the questions will come very quickly. Right now, we don't have any in the chat, but I'm sure people are writing them up. So I have the first questions for you. Like with climate change coming in, do you think you will have like an increase in biodiversity in your area? For example, what will be the effect? Because in the end, it's a change of condition, a bit like tectonics, I guess.
Yes. Actually, there are several hypotheses. So people look the climate from last glacier maximum to now, and then they found actually the regions with low climate velocity will have a higher richness. That means if a region has more stable climate, that will maybe allow species to persist in such region. And if the region with a very big shift of the climate, maybe species cannot catch the speed of climate change, and they finally got extinct.
Okay, yes. Thank you very much. We have a first question in the chat. Yichi is asking, may I ask when during the geological history you think there was a change in the number of plant species in the Hengduan Mountains?
Sorry, can you,
So in the last geological history, when do you think there was a change in the number of plant species in your area?
during the geological history, so I guess maybe it represented like the intensive uplift in the Hengduan Mountains. I think the uplift will definitely can increase the species richness because it could provide novel habitat for species to colonize and diversify. And if they have a more disrupt landscape, it tends to have higher species richness. But how to quantify them is still kind of a big challenge.
Okay, thank you very much. Maybe Pedro or Tamara, do you have questions? Because I can continue, but...
Yeah, so we're still waiting for more questions in the chat room. But Yaquan, thank you very much. This was very inspiring. And I wanted to ask you, you looked at climate reducing or controlling for the climatic factors to then look at potential topographic or tectonic factors. But I was wondering about the effect of other species like primates or other living beings, birds, bees, etc., as dispersal agents to drive diversity. And so I was wondering if you have any thoughts on controlling for these other biological factors that can help increase plant diversity.
Yeah, I see your point. So you mean for these biotic interactions to see how they impact on the species richness pattern. Well, I mean, it definitely has big impact, but it's also a scale dependent question, if I understand correctly. If you look more into the local scale, I think about biotic interactions may play a larger role. But if you look at the regional scales and this compared to the climatic and geomorphic impact, this part maybe can play a very small role. But I think it's good to take in consider of it. And I know some landscape evolution model are also thinking this way. For instance, there is a landscape evolution model called ADAScape. So it's developed from FastGape, and there they are using the different processes. And in their model, they include the computation process to see how it could drive the biodiversity patterns. Yeah.
Okay, we have a question from Luca. So it says, thank you, Tron. Do extensive glaciations, like in the European Alps, erase the board and a new local diversity is established at each deglaciation? Or do some species, like fungi, survive? And then there's a following question. Is there shorter time scale since deglaciation enough to establish a speciation?
Thanks. I think for this question, glacier definitely has a huge impact, and here we know the LGM or the climate, the glaciers has a big impact, especially in the Europe, and because of the glaciers, a lot of species just has been erased, and I think in the interglacial cycle, and the species could colonize back, but there are some studies said even until now, the species are still in the colonization process. So they are still not reached the full equilibrium of the landscape. So that means there are still a lot of space for species to colonize. And also, and here I would also like to mention that the direction of mountain range also plays a big role, because the Alps is east-west oriented, and during the glaciations, like the species cannot move south because the Alps just act as a barrier. But for instance, in the US and in Southeast Asia, these mountain ranges are north-south oriented. So in this glaciation period, species actually easier to migrate southwards to escape from the glaciation areas. Yes. So here I would like to say, like, we are especially in the Europe, like it's still in this equilibrium stage, and the species still need time to establish their population.
So we definitely like have a difference between geological input and climatological input. We have Yelena in the chat that is asking, when do you expect to publish this work? I think she's very impatient. And in which journal?
Well, for my PDA studies, I hope I can submit it very soon. And I hope I can submit it in like 1 or 2 months, because it's more or less ready. And for these realm studies, and I'm still working on it, because like the signal is still kind of not that stable, and maybe I can submit in half a year. And in terms of the journal, I would like to submit to these cross-disciplinary journals, and then to advertise more to the geologists. Yeah.
Thanks, that would be very interesting. Well, I have a question that was a bit more technical, I guess. Maybe Tamara, you want?
Oh, yeah, sorry. I had a question. My unmute was not working very well, but yeah, I this was super inspiring too. Very, very interesting. And my question is related to how shorter term processes can impact the biodiversity, because these catchments can be affected, considering these faults are really active and had multiple earthquakes that are triggering, I don't know, thousands of landslides, and how this massive occurrence of landslides can impact the biodiversity and the recover of the species. I don't know if you have any thoughts on that.
Well, actually, I don't have a very clear idea on it, but I think for these short scale geological processes, it might impact on the species diversity at the population level. Maybe you are not be able to identify this is already a new species, but they might be like, say, they are different populations. And this type of study is another field called population genetics. And I think usually like the speciation rate, like per million years, maybe like 1 or 2 species. So the developing of new species is pretty slow, and it's also taxa dependent. Like for maybe mammals, maybe takes much longer time to let them speciate, but for maybe for plants, maybe it has much shorter time. Yeah.
Thank you very much. Also for Yelena, yeah, we have now your past articles in the chat. I was wondering, like, at some point you mentioned that the DNA, you had like this shift between like the elevation distribution in one catchment, and then the distribution, I think, of elevation that you got from your samples, I guess. Is there a way like to compute beforehand, so before you can go in the field and you measure that, do you have like any idea of how to calculate, like have an idea of it will work, and you will get the whole elevation range? Like, is it linked to DNA survival in water or something?
Yeah, it's possible, and according some hydrological conditions and maybe water speed and the taxa you focused, I think there are some models to working on it, and they can just to sample to get the detect possibility for the whole drainage basins and to help you to optimize the sampling strategy. I think the Professor Florian Atmod group in the University of Zurich developed these kind of models. And here, because I'm a kind of modeler, and I didn't think that much to how to develop a very good fieldwork strategy. And I just think, yeah, the geology using this basin average erosion rate, maybe I can just go to the outlet and then I can sample the whole drainage basins. But in fact, I could not. But it's also good that if I just look at the mid to low elevation species, I could consider the drainage divide is a dispersal barrier. Or if I look at the maybe high elevation species, maybe the river valley is a dispersal barrier. So my current approach, it's also kind of good to account for the habitat preference, but there is definitely a way to optimize the sampling strategies.
Thank you.
All right, we're short on questions in the chat. I'll ask mine, and then we can see if Tamara has any more questions, and then we'll let you go. I was wondering in your if you came across maybe you showed this and I missed it, but about the ages of these species within the valleys, and if you can accurately determine those from, you know, the phylogenetics or something like that, and if those ages, you know, how old are these species, and are they dependent on, like, really dependent on the geological evolution of these uplifting valleys, right?
I see. Well, here you mean this figure, right? So shows the tape age from phylogeny.
So that's about 1.5 million years for these species that you're working with?
No, it's just a toy example.
Okay.
So I just to show the concept of how we get the tape age. And here we just to measure for the existing species, we can say nowadays and how old they are. And this just considers the last divergence time, and to say if they have a lot of old species living there, and to correlate the exact timing with the erosion history. I think maybe this is not a very good approach, because it's just consider the last split. And another way we are trying to do is to look at the full evolution history of the phylogeny, and to say when they have a higher speciation rate.
Right.
Yeah, using this type of information, we can compare with the erosion history in a more clear way, Yeah.
Yeah, that's
So
fascinating.
yeah.
I was just and related to the age question, I was wondering about, you know, how accurately you can determine the age from these molecular clocks, because they tend to be pretty you know, the uncertainty on them tends to be pretty high, but maybe it's a bit different for plants.
Yes, I see your point. I didn't make this phylogeny by myself, and I just use this published phylogeny. And the phylogeny I choose is at the clade level. So I just choose several clade that are highly diversified in the Hengduan Mountains. So, and then like for this type of phylogeny, they are not collect species within the Hengduan Mountains, also around the world to see all the species within this clade. And using this type of phylogeny, the age information, I would say more or less more precise, because if you use this kind of regional phylogeny, they just get all the species in this region and get the phylogeny. Maybe the age is not that precise, because maybe there are other split in the phylogeny, but the species occur in the other places in the world, but you cannot track it from the phylogeny. And I think I have like a very complete table about the quality of this phylogeny, and I think that's the best I can do so far. Yeah.
Great. Thank you. Yeah.
Okay, thank you. Tamara, do you have a last question?
I do have a last question if nobody in the chat is asking, but maybe you said that and I could have missed it. But for the river, the Yangtze, I think was the river that didn't act as a barrier, the ridge between yeah, it's like on the second part. Right. So you show us that this one, the second one that doesn't act as a dispersal barrier. I was wondering, you said something that was tax dependent, but is there any other reason that you might think that this second divide is not acting as a dispersal barrier? What characteristics about that, maybe geological or biological, can be controlling that doesn't act as a barrier?
Yes, I'm still thinking about the reason. And something in my mind is in this Southern Mekong Divide, the relief is much higher. And from this Mekong-Yangtze Divide, there is a drainage divide, and they are pretty wide, but actually the relief is not as high as the Southern Mekong Divide. So that might be one reason. It is because the divide is steeper and it will increase the difficulty to cross the divide. And another thing, I don't know if I should believe it or not, is we are also thinking about the divide migration process. And here in the Southern Mekong Divide, it consistently moved eastwards. But for the Mekong-Yangtze Divide, like some places move eastwards, some places move westwards. Maybe that created like the mix of the species, but I'm not super sure about it. And if you want to know more about the tectonic and geomorphic patterns of this region, my colleague Katrina Gelwick published a paper in eSurf talking about this geomorphic matrix. Yeah.
Great. Thank you.
Thank you, Yaquan. It was a super interesting talk, so I think we can finish now. And yeah, just the next week we also have a talk from Sinead Liste on the impact of vegetation on delta landscapes and stratigraphy.